welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're understanding what is meant by passive transport, active transport, endocytosis, and exocytosis. We'll also be looking at the involvement of carrier and channel proteins in membrane transport. So diffusion occurs across the cell membrane, and it is the net movement of a substance from a region of its higher concentration to a region of its lower concentration. And net movement refers to the overall direction of movement. The molecules go down the concentration gradient. Diffusion will continue until equilibrium is achieved. This is when the concentration of substances are the same on both sides. Small and non-polar molecules can easily diffuse directly through the cell membrane, and this process is called simple diffusion. Small molecules can fit through the phospholipids, which is what cell membranes are made of, and non-polar molecules can interact with non-polar tails of the phospholipids, which is why non-polar molecules can pass through cell membranes, as they are not repelled by the hydrophilic head of the phospholipids. So the rate of diffusion is affected by various factors, and they are concentration gradient, temperature, surface area, and properties of substance involved. So large polar molecules cannot pass through cell membranes directly, and they need the support of transport proteins to pass through the phospholipid bilayer. This form of diffusion is known as facilitated diffusion. Facilitated diffusion is possible through two types of transport proteins, and these are channel proteins and carrier proteins. These proteins are very specific and only allow one type of molecule or protein to pass through. Channel proteins are pores that run right through the membrane. They allow the passage of charged substances such as ions. Most channel proteins are gated, hence giving more control to the channel protein over what ions can be exchanged. So now we're looking at carrier proteins. So carrier proteins can switch between two shapes, in contrast to channel proteins, which has a fixed shape. So channel proteins are almost like a tunnel that you go through, while carrier proteins are like gates. So that's one way to think about it. So the carrier protein is open on the side so that the ion can be transferred can enter, and then it opens on the other side for the ion to be released. The direction of movement diffusing depends on the concentration gradient once again. So what is active transport? So active transport is the movement of molecules and ions through a cell membrane from a region of a lower concentration to a region of higher concentration. So the active transport goes against the concentration gradient, hence it requires energy in the form of ATP. Active transport takes place in carrier proteins, and energy is required for the carrier protein to change shape. So we just looked at how carrier proteins work. They need to change shape so that the molecule or whatever needs to be transferred enters the cell membrane first, and then it needs to pass the carrier protein. So that's why it needs energy to change shape. ATP is hydrolyzed to release the energy needed. Examples of active transport. So these are probably examples you've come across in GCSE as well, but then it's quite good to refresh because they often come up as 1 markers. So reabsorption of useful molecules and ions into the blood after filtration in the kidneys is one example. Absorption of products of digestion from the intestines into the blood, loading sugar from the leaves into the phloem tissue for transport. So that's more to do with topic 5, which has already been uploaded. So do check it out if you need to. And finally, absorbing inorganic ions from the soil into root hair cells. Cytosis. So some molecules are too large to travel through the membrane. And some examples of these large molecules are proteins, lipids, and carbohydrates. Hence, to absorb them, because they are very important, a cell surrounds a substance with a section of the cell's surface membrane. So when they do that, when the cell does that, a temporary vacuole is formed with the ingested substance inside, and the membrane engulfs the substance. So this is an active process and requires energy. So what about the opposite, exocytosis? So some substances, such as hormones, enzymes, and lipids, need to be secreted. Hence, vesicles containing the substance is pinched from the sacs of the Golgi apparatus. So this will be explored in more detail in topic 3, and which will soon be uploaded, so do not worry. But yes, all you need to know for now is that vesicles containing the substance is pinched from the sacs of the Golgi apparatus, and these vesicles move towards the cell surface and fuse with the cell membrane. So it's basically the opposite of endocytosis almost. And the contents of the vesicles are released, and this process is called exocytosis. This, just like endocytosis, is also an active process and therefore requires energy. So that is all from Professor Snap today. Hope you found this video useful. If you have any questions, please let me know in the comments below, and don't forget to like, share, and subscribe.